Hi everybody, so today we're going to be going over chapter 4, which is titled Carbon and the Molecular Diversity of Life. So all living organisms are made up of chemicals that are based mostly on the element carbon. And carbon is able to enter the biosphere through the actions of plants and other photosynthetic organisms, which use solar energy to transform carbon dioxide into organic molecules, which are then taken in by plant-eating animals. And of all of the elements that exist, carbon is unparalleled in its ability to form molecules that are large and complex and just form molecules that make possible the wide variety of living things. Proteins, DNA, carbohydrates, and other molecules that distinguish living matter from inorganic material are all composed of carbon atoms that are bonded to each other and to atoms of other elements. And these other elements commonly include hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So before we're able to define organic chemistry and organic compounds, we need to go over the history of how we got to these definitions. So early chemists proposed this concept of vitalism. And vitalism, as a concept, it actually originated during ancient times, and it has been seen in the work of philosophers such as Aristotle. But it was not until the 17th to the 18th century that vitalism began to gain traction due to experiments by certain chemists such as Becker and Stahl. And what this concept aimed to do was distinguish life from non-life. And it claimed that inorganic materials were directly derived from non-living sources, such as minerals, salts, and metals. And on the other hand, it claimed that organic materials were derived from living sources, such as plants and animals. This concept said that all life was governed by an unexplainable force, and that the laws of chemistry and physics did not apply to life. But eventually, in the 19th century, a chemist named Friedrich Wöhler was able to synthesize urea, which is a living compound present in animal urine, from ammonium cyanate, a non-living salt, and he synthesized this compound with heat. And this reaction, it's now referred to as Wöhler synthesis, and this reaction proved that organic compounds can arise from inorganic precursors. And ever since Wöhler's discovery, organic compounds were now defined as compounds that contained carbon and hydrogen bonded to each other, and inorganic compounds were defined as compounds that generally did not contain carbon bonded to hydrogen. And while there were other experiments that did challenge the concept of vitalism, before this experiment, a very important one that happened that was able to finally discredit it fully was Miller's experiment. So basically, in 1953, there was a chemist and his name was Stanley Miller. And what he did was he set up a laboratory simulation of the possible chemical conditions that occurred on primitive Earth. And what he was able to do was demonstrate that there was a spontaneous synthesis of organic compounds. So what he did was he recreated the early Earth's atmosphere in a closed system that included a flask of water that represented the ocean. He then heated this flask and produced water vapor. And this water vapor entered a chamber that contained gases such as methane, ammonium, and hydrogen, as these gases were believed to have been present at that time on Earth. And then there were electrodes that simulated electrical sparks that were discharged into this gas chamber. And that was the source of energy. It simulated lightning, and that lightning is what would drive chemical reactions in the atmosphere. And after passing through the gas chamber, the vapor cooled in a condenser, which caused the new compounds that were formed to dissolve into a liquid, and as a result, the liquid would be trapped at the bottom of the system. And periodically, he would analyzed these samples, and what he found was that these samples, they contained amino acids, which are the building blocks of life, and other organic compounds. And effectively, that's how this experiment was able to disprove vitalism once and for all. And although his experiment was a success, the mixture of gases that Miller created probably did not actually accurately represent the atmosphere of the primitive Earth. But similar experiments using more accurate atmospheric conditions also led to the formation of organic compounds, which just proved the concept of abiotic synthesis. And it's thought that spontaneous abiotic synthesis is what happened in early Earth, and it may have occurred near volcanoes. And this abiotic synthesis is just the spontaneous generation of organic compounds from inorganic compounds. And they think it occurred near volcanoes because near volcanoes is the area that would have provided the right combination of different things such as heat, minerals, and gases that would facilitate the chemical reactions that were necessary for the formation of these amino acids and other organic compounds. So it is thought that the spontaneous abiotic synthesis of organic compounds may have been an early stage in the origin of life on Earth. And from analysis from experiments such as Miller's experiment, we were able to find out that the overall percentage of the most fundamental elements of life, which are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, they were all uniform from one organism to another. And this also is just reflecting the common evolutionary origin of all life. And because of carbon's versatility, these few elements can be combined to build an inexhaustible variety of organic molecules. So now that we've discussed the history of organic compounds and organic chemistry, we should talk about how organic chemistry is defined. And basically, organic chemistry focuses on organic compounds that contain carbon. And this definition of organic chemistry arose because of Wöhler's synthesis, which proved that organic compounds came from inorganic compounds, and Miller's experiment, where he demonstrated spontaneous abiotic synthesis. And organic chemists finally rejected vitalism due to experiments like these, and they were able to embrace the idea of mechanism. And mechanism is just the belief that the same physical and chemical laws govern all of natural phenomena, including life. So, organic chemistry first began with attempts to purify and improve the yield of products that were obtained from organisms. And while these chemists were able to synthesize simple compounds, they had difficulty with more complex ones. So Wöhler's synthesis, Miller's experiment, and other key experiments demonstrated the versatility of carbon and bonding, which is what leads us to our next concept. The idea that carbon atoms can form diverse molecules by bonding to 4 other atoms. So because of carbon's ability to form 4 bonds, it is the backbone of biological compounds. And the bonding patterns it has is what allows for carbon to create a diverse group of structures such as chains, rings, and branch networks that are the foundation of our proteins, nucleic acids, carbohydrates, and proteins. So to understand its bonding patterns, we first need to understand its electron configuration. So to begin, carbon has a total of 6 electrons, 2 in the first shell and 4 in the second shell. And we know that the second shell of an atom can hold up to 8 electrons. And what this means is that carbon is going to have a little tendency, very little tendency, to form ionic bonds, which is the losing or gaining of electrons to complete a valence shell. Instead, carbon usually completes its valence shell by sharing electrons with other atoms in the manner of 4 covalent bonds. And these bonds may include single or double bonds. So the ability of carbon to form 4 covalent bonds is what makes large complex molecules possible. So some of the different bonding patterns that carbon can exhibit are on this slide. And basically everything on this slide is a simple hydrocarbon. And hydrocarbons are just carbons and hydrogens that are bonded to each other exclusively. So when a carbon atom forms covalent bonds with 4 other atoms, they are arranged at the corners of an imaginary tetrahedron with bond angles of 109.5 degrees. And these tetrahedrons include methane and ethane. And in methane, we see that we have 4 hydrogens, and these hydrogen atoms are positioned they're symmetrically positioned around our central carbon. And we need to remember that our molecules have a 3D shape, and that the bonding angle of methane is going to be right here, and it's going to be 109.5 degrees. And when we have a molecule such as ethane, which has multiple carbons, every single carbon is going to be bonded to 4 other atoms, and it is going to maintain the tetrahedral shape. So if we take a tetrahedral structure like this one that we see here, and if we are to take it and we change its positioning like this, and we turn it, what we get is 2 tetrahedrons together, even though our molecule has one more carbon than the previous one. And that's how we get ethane structure. So no matter the molecule, as long as carbon and hydrogen are bonded with a single bond, it is going to adopt this tetrahedral shape around each carbon. But what happens if the molecule that we're talking about bonds carbon with a double bond? Well, when we're looking at ethene, we see that there is a double bond between our 2 central carbons here. And what happens is that the shape we're going to get is actually called a trigonal planar. And this has a bonding angle of 120 degrees. And the reason for this is because this double bond right here in ethene causes the molecule to have a flat planar structure, and that does not allow it to have any rotation from the carbon to carbon at the carbon to carbon double bond. So when it comes to bonding patterns of an element, we've talked about these bonding patterns being determined by an element's valency. So an element's valency is going to determine the predictable number of bonds that are formed. And if we remember, valence is just referring to the atom's outermost shells and electrons. And those valence electrons are the ones that are able to interact with other atoms and form bonds. So when we're looking at carbon, we see that carbon has 6 electrons. 1, 2, 3, 4, 5, and 6. There's 2 in the first shell, and there's 4 in its valence. And since the second shell can hold up to 8 electrons, carbon is usually going to share electrons with other elements to complete its valence shell, meaning that carbon's valence is 4, and that it can share its electrons with other atoms so that it completes its shell, meaning that there are 8 electrons in it. And that is actually what the octet rule states. It states that there is a tendency of atoms to prefer to have 8 electrons in their valence shell because it is actually more energetically favorable. So because carbon can make 4 bonds, it is called tetravalent. So when we look at nitrogen, we see that nitrogen has 7 electrons, which is 1, 2, 3, 4, 5, 6, 7. We have 2 in the first shell and 5 in the second shell. So to satisfy the octet rule, nitrogen is going to form 3 bonds, which makes it trivalent. And an example of a triple covalent bond like we would see here with nitrogen is it's kind of like this. We have our nitrogen. It has 7 electrons, 1, 2, 3, 4, 5. And only 5 of those 7 are in its valence shell. So when we have N2, we have 2 nitrogens with their 5 valence electrons. And when we add them together, what we get is this structure. A nitrogen with 2 valence electrons, another nitrogen with 2 valence electrons, and then a bond between those 3 electrons that we see here. And this is what's called a triple bond. So these 3 electrons bonding causes it to have a triple bond. And this same concept is going to apply to oxygen. We know that oxygen has 8 electrons, 2 in the first shell and 6 in the second shell. So it has an ability to form 2 bonds to complete its octet, meaning that it's divalent. And because oxygen has 6 valence electrons, they're going to share 2 pairs of electrons and create a double bond between them. So when we have O2, we have 2 oxygen molecules. They have 6 valence electrons. And what happens with these is that we're going to have a sharing of these 2 electrons. So when we share them, it looks like this. And to simplify it, it's actually going to look like this. So hydrogen has 1 electron, and it forms 1 bond to complete its valence, which makes it monovalent. I also did want to note that hydrogen, oxygen, and nitrogen are the most frequent bonding partners of carbon. And another important note that I need to make is that while atoms do have the ability to share electrons, they can also donate or lose electrons. And when they donate or lose these electrons, they're going to obtain a formal charge, and then they are going to be called ions. So formal charges are associated with atoms that do not exhibit the appropriate number of valence electrons. And when we have an atom that gains an electron, it's called an anion. So an anion is equal to a gain of electrons. And this is because it now has a more negative than positive charge because there are more electrons and protons in the atom. And when an atom loses an electron, it's called a cation. Loss of electrons. And cations are always going to be positive because now the atom has more protons than electrons. So for the next section of this lecture, we have molecular diversity arises from variations in the carbon skeleton. So we need to remember that molecules are 3D structures and that their shape is imperative to their function. Form always follows function. And one of the very important characteristics that carbon has is its ability to form carbon chains. And carbon chains are a series of carbon atoms that are bonded together. And these carbon chains are going to form carbon skeletons, which are the backbone of many organic molecules. Actually, they're the backbone of most organic molecules. So carbon chains can be connected to each other by single bonds, double bonds, or triple bonds. And normally, hydrogens or other groups that are attached to them that complete their valence. So carbon skeletons, they can greatly vary in length. We can have a compound like we see here, which is ethane, that only has 2 carbons. And we can also have compounds like polymers, in which its skeleton can have hundreds or even thousands of carbons. And carbon skeletons, they can be branched, like we see here with 2-methylpropane, or they cannot be branched, like we see with butane. They can be straight. They can even be arranged in rings, like we see in cyclohexane and benzene. And they can even have double bonds. They can actually even have triple bonds as well. And all of these variations, they are important sources of the diversity that characterize living matter. They undergo reactions and they release a large amount of matter. And these hydrocarbons, they can be found in places like petroleum. That's an example of a hydrocarbon. They can be found in molecules such as fats. And fats, they tend to have long hydrocarbon tails that are attached to a non-hydrocarbon component. And even though we normally see hydrogens attached to our carbon skeleton, we do have other atoms or functional groups that can bond to the skeleton, and that forms an even larger variety of chemical structures and functions. And another way that we are able to see hydrocarbons is as isomers. And isomers are able to build on hydrocarbons by introducing variations in the arrangement of their atoms. So isomers are compounds that have the same molecular formula, but they have different structures and properties. They have the same number of atoms of the same elements, but they have a different structure and function. And we can think of isomers as a variation of the architecture of organic molecules. And one type of isomer we have is known as a structural isomer. They're also called constitutional isomers, and what they are is that they are molecules that have the same formula, but they have different covalent arrangements of their atoms. For example, in pentane and 2-methylbutane, they both have 5 carbons and they both have 12 hydrogens. So their molecular formula is both C5H12, but their differences are in their molecular shape, which is going to lead to differences in their function. We see here that pentane is linear, and we see here that 2-methylbutane is branched. And we need to remember that form always follows function, and changing the form is going to result in a change of function. So pentane is actually known for its use as a solvent and as a component in fuel. Meanwhile, 2-methylbutane, which is also known as isopentane, it's known for its role as a gasoline additive, and it's also known as a cryogenic agent for quick freezing in laboratories. Another type of isomer we have is known as cis-trans isomers. And cis-trans isomers are also called geometric isomers. They have the same number of covalent bonds, but they have a difference in spatial arrangements. And these differences are going to rise due to the restriction of rotation around this double bond, around this carbon to carbon double bond or ring structure. And the inflexibility of these double bonds is what prevents the atoms from being able to rotate freely, which is going to lead to distinct shapes and properties. So in cis isomers, like this one, we see that the two X's are on the same side. Meanwhile, in our trans isomers, our X's are on opposite sides. And since form follows function, it's only natural for cis and trans isomers to have different characteristics. So cis isomers are known for having chemical reactivity, and they interact with enzymes or receptors. Meanwhile, our trans isomers, they have different properties. They're normally more stable than cis isomers, and they usually have a higher melting point than cis isomers. So the last type of isomer that we have are called enantiomers. And enantiomers are isomers that are mirror images of each other, but they cannot be superimposed. So we cannot perfectly place enantiomers on top of each other. It's sort of like how our hands are mirror images of each other, but they can't be perfectly aligned. And enantiomers are possible when 4 different atoms or groups are bonded to an asymmetric carbon. So they are possible due to bonding to asymmetric carbon. And because form follows function, and even though enantiomers are chemically the same, they behave different in the body because form follows function. One enantiomer might be active and have a desired effect, meanwhile the other one can be inactive or cause side effects. And a common example of enantiomers includes L-dopa and D-dopa. And an L isomer is an enantiomer, stands for levo, which stands for left, and D, which stands for devo, which means right. So basically, L-dopa is able to effectively cross our blood-brain barrier, which makes it effective in treating Parkinson's disease. However, D-dopa, it cannot cross that blood-brain barrier, so it can't treat Parkinson's. So they have different effects even though they are the same chemical structure. And having a strong understanding of enantiomers is necessary for being able to design drugs and treatments that not only do they have to be effective, obviously, but they need to be safe. And this is why enantiomers are so important to the pharmaceutical industry, because even a small difference in their 3D structure can lead to a very different biological outcome. So the next concept of this chapter is that a few chemical groups are key to molecular function. So far, we've said that what makes an organic molecule unique or gives it its distinct properties is the arrangement of its carbon skeleton. However, there's another key factor that contributes to the uniqueness of a molecule, and that is its chemical groups. So, chemical groups are attached to our carbon skeletons, and these groups, which can normally also be called functional groups, they are specific combinations of atoms that affect the molecules or chemical behavior, its reactivity, and its interactions with other molecules. And these chemical groups, they may be involved in chemical reactions, or they can contribute to the shape and function of the organic molecule in a characteristic way which gives it its unique properties. And a common example of this is actually the basic structure of testosterone, which is a male sex hormone, and estradiol, which is a female sex hormone. And what we see is that both of these hormones, they are steroids that have 4 fused carbon rings. But these hormones, they differ in the chemical groups that are attached to the rings. So in estradiol, we have an OH group, which is known as a hydroxyl group. And this hydroxyl group is attached to one of the rings, which is what makes it an estrogen hormone. And this hydroxyl group, it contributes to estradiol's ability to interact with specific receptors in the body, influencing its function in the regulation of reproductive and other biological processes. But on the other hand, we have testosterone, which is another steroid, and it has a CO group and a methyl group, which is CH3, and this is our CO group. And our CO group is just known as carbonyl group. And our CH3 is known as a methyl group, with our OH being our hydroxyl group. And the fact that testosterone has a carbonyl group and a methyl group attached to it is what gives it its distinct properties and effects, such as the promotion of the development of male characteristics. So the differences in functional groups of a molecule is what's going to give that molecule its unique biological activity. And in other cases, functional groups, they affect molecular function through their direct involvement in these chemical reactions. And the term chemical groups and functional groups, they are often used interchangeably, but functional group refers to a specific group of atoms that are attached to a molecule that influence its chemical reactivity and behavior. So what we see here are functional groups being involved in chemical reactions. Meanwhile, our chemical groups is a more general term that can include any group of atoms, which includes functional groups. So we have a total of 7 chemical groups, which are important to the chemistry of life, and they are our hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, and phosphate group, and our methyl group. And these first 6 groups, they are hydrophilic, and they increase the solubility of organic compounds in water. And it's important to know that methyl groups are not usually reactive, but they do serve as very important markers on our organic molecules. So now we're going to go over some of the properties of these functional groups. So in a hydroxyl group, we have a hydrogen atom that forms a polar covalent bond with an oxygen atom, which forms a polar covalent bond to the carbon skeleton. And this is because these polar covalent bonds of hydroxyl groups, they actually increase the water solubility of these organic molecules. And organic compounds with hydroxyl groups are called alcohols, and they usually end in ol, O-L. So an example of an hydroxyl group would be ethanol. So here we have our central carbons, we have 2, and surrounding it we have our hydrogens. And on the other side, we have our hydroxyl group here, and we see that this hydrogen atom is forming a polar covalent bond with this oxygen because oxygen is more electronegative than hydrogen. And then this oxygen atom forms a covalent bond with this carbon skeleton. A carbonyl group is going to consist of an oxygen atom that is joined to the carbon skeleton by a double bond. So an example of this would be acetone, right? We have a carbon, a hydrogen, another hydrogen, hydrogen. And then we have another carbon, another one. We have our hydrogens. And here in this central carbon, we're going to have a double bond to the oxygen atom. And if the carbonyl group is on the end of the skeleton, it is going to be called an aldehyde. But if the carbonyl group, like we see here in acetone, if it is within the molecule, if it is within the carbon skeleton, it's going to be called a ketone. So the next group that we're going to go over is the carboxyl group. And a carboxyl group, it consists of a carbon atom that has a double bond to an oxygen atom and a single bond to the oxygen atom of a hydroxyl group. So an example of a carboxyl group is going to be acetic acid, right? So we have carbon, hydrogen, hydrogen, hydrogen. These are single bonded together. We have our next carbon with a double bond to our oxygen and a single bond to our hydroxyl group. So of acetic acid, our carboxyl group is going to be this section right here, where we have a double bond between a carbon and an oxygen and a single bond between a carbon and a hydroxyl group. And compounds with carboxyl groups, they're known as carboxylic acids or organic acids. And a carboxyl group is able to act as an acid because of the combined electronegativities of the two adjacent oxygen atoms that increase the dissociation of hydrogen as an ion. So the next group that we're going to talk about is an amino group. And an amino group consists of a nitrogen atom that is bonded to two hydrogen atoms and the carbon skeleton. So for example, we have glycine, okay? So we have a hydroxyl group. It is bonded to a carbon, and then this carbon is double bonded to an oxygen. And what did we just say? We said that anything that has a single bond to a hydroxyl group and a double bond to an oxygen that is connected to a carbon skeleton is actually a carboxyl group. So here we're seeing a carboxyl group, and this carboxyl group is going to be bonded to a carbon that is bonded to our hydrogens. And then this carbon is actually going to be bonded to a nitrogen and then two hydrogens. So this is our amino group. So organic compounds that have amino groups are known as amines. And this amino group is actually able to act as a base because it has the ability to pick up hydrogen ions from a solution. And amino acids are just the building blocks of proteins, and they have both amino and carboxyl groups. Another group that we're going to be talking about is a sulfhydryl group. And this group, it's going to consist of a sulfur atom that is bonded to a hydrogen atom and the carbon backbone. And these groups, they normally are going to resemble a hydroxyl group in shape. And the organic molecules that have sulfhydryl groups, they are known as thiols. So two sulfhydryl groups can actually interact and help to stabilize the structure of proteins. Another group that we're going to talk about is our phosphate group. And our phosphate group contains a phosphorus atom that is bound to 4 oxygen atoms, 3 with single bonds and 1 with a double bond. So for phosphate groups, we have glycerol phosphate. And what we see with glycerol phosphate is that we have 3 carbons they have single bonds to hydrogens. The carbon skeleton is bonded to itself. We have hydroxyl groups. And then we have our phosphate group, in which we see that a carbon in the carbon skeleton is going to be bonded to an oxygen, and this oxygen is going to be bonded to our phosphorus. It's going to be bonded to two more oxygens with a single bond, and then it's going to have a double bond to one of the oxygens. And the phosphate group is connected to the carbon skeleton via one of its oxygen atoms. And phosphate groups, they are actually anions with two negative charges, which is what we would see here, because the two protons dissociate from oxygen atoms. And one function of phosphate groups is to transfer energy between organic molecules, and it does so by the energy being ATP. And lastly, we have our methyl group. And a methyl group is just a carbon that has three hydrogens bonded to it with single bonds. And the addition of a methyl group to certain molecules like DNA can silence gene expression through a process called methylation. And methylation typically is going to prevent a gene from being transcribed and expressed. And methyl groups actually also have the ability to affect the shape and function of our sex hormones, such as estrogen and testosterone, and it does this by influencing how hormones bind to receptors and interact with target cells, which is going to alter their biological effects. So the last concept of this chapter is that ATP is an important source of energy for cellular processes. So we just talked about our functional groups, and we learned that our functional groups are going to play a vital role in the structure and function of biological molecules. And these functional groups are going to influence their reactivity, the solubility, and their interactions. And among these functional groups, the phosphate group is actually particularly significant because it is a key component of our molecule adenosine triphosphate, which is also known as ATP. And ATP is normally known as the energy currency. So ATP is the energy currency of our cells. And ATP relies on high energy bonds between the phosphate groups to store and transfer this energy. So ATP is the primary energy transfer molecule in living cells. And ATP consists of an organic molecule called adenosine, which is what we see right here, and ribose, which is what we see here. And adenosine and ribose together is going to be attached to a string of 3 phosphate groups. And adenosine is formed by adenine and ribose. And this adenosine is going to be connected to our phosphate groups. And these phosphate groups are linked by high energy bonds, and they are particularly reactive. And when one of these bonds is broken through a reaction with water, an inorganic phosphate ion is going to be released, which is inorganic phosphate ion, and its ATP is going to become adenosine diphosphate, or ADP. And the process by which a bond within a molecule is broken by the addition of a water molecule, this is known as hydrolysis. And during hydrolysis, a water molecule is going to split up into a hydrogen ion and a hydroxide ion, and then they are both going to be incorporated into the products of the reaction. So when we think about it, our ATP is able to store the potential to react with water, which is what makes it essential in in the molecular bridging between chemical structure and cellular energy. And this is just highlighting the critical importance of our phosphate groups as a functional group, and how as a functional group it has a broader role in life's biochemical processes and chemical reactions.